Modern histologists recognize two major cell types in the central nervous system: the electrically excitable neurons and the neuroglial cells, variously described as connective tissue cells, nutritive cells, or phagocytes. The term neuroglia was coined by Virchow in the 1850s. In this original illustration of a section of the brain, he describes neuroglia as the material between the cells and regarded it as a glue holding the neurons together. Between the middle of the 19th and 20th centuries, staining systems, mostly using salts of the heavy metals, particularly silver, were developed by Golgi, Ramon y Cajal, Del Rio Ortega, and many others. These were used to characterize different kinds of cells in the central nervous system by light microscopy. By the early 1920s, the currently accepted view had evolved that neuroglia was not a glue or a ground substance, but a tissue composed of three types of cell: astrocytes, oligodendrocytes, and microglia. Each type requires specialized staining systems for them to be seen. Astrocytes, illustrated here with drawings by Penfield, have long fibrous processes, often ending on capillaries. Oligodendrocytes are pictured, again by Penfield, as slightly oval cells with fewer processes. Baumann and Bock used Rio del Ortega's stain to show a microglial cell, which is described as having a small dense nucleus and a slightly staining cytoplasm. It appears in different publications in many forms, but is generally considered to be phagocytic. It is interesting to note that in the literature, actual photomicrographs of neuroglial cells are much rarer than diagrams or drawings of these cells. In fact, the literature offers no universal agreement on the cellular structure of the central nervous system. For example, here are some adjectives from authoritative publications used to describe neurons. Similar publications describe oligodendrocytes thus, and microglia thus. Descriptions of astrocytes are very rare.
Perhaps the main reason for a lack of an agreed histological view is that our knowledge of the brain is so fragmentary. But there are other more practical difficulties contributing to the confusion. And it is important in this context to look at three areas in some detail. These can be discussed under the following headings.
Firstly, the appearance of a cell in any particular section depends mainly on the orientation of the cell in relation to the angle of the knife, and the thickness of that section relative to the dimensions of the cell body. This can be demonstrated by considering the solid geometry of an egg as a model of a large cell body and its nucleus. We can cut slices of the egg with the same relative thickness as a neurohistologist uses in preparing sections for staining. Here we see three eggs of the same size that have been sectioned along three different axes, resulting in a large variety of two-dimensional sizes and shapes, despite the uniformity of the uncut eggs. The more asymmetrical the cell body, the more different kinds of appearances would be seen. Quite clearly, one cannot describe the size, presence or position of the nucleus, nucleolus or mitochondria, or their shapes, without cutting serial sections. This difficulty is compounded if we are looking at cells like neurons that have processes. How can we be sure of the numbers and positions of axons or dendrites? If we are to distinguish between cell types on the basis of appearance, then having a clear understanding of their three-dimensional structure is even more critical. In the case of the central nervous system, this point is illustrated by these beautiful drawings of Peters, Paley, and Webster, showing neurons, an oligodendrocyte, and an astrocyte. It is obvious that the external structure of the different cell types drawn by these distinguished neurohistologists would be interchangeable if any of these cells were rotated and viewed from a different angle. Secondly, any staining procedure alters cells considerably and distorts their organelles. These are two unfixed neurons isolated by hand dissection. You can see that they are fairly globular with large nuclei. Watch what happens as they are fixed and stained. This sequence was recorded in real time and then edited to reduce the overall running time on the screen. We may now compare the final appearance with the original appearance of the same cells. Thirdly, the different staining systems for the different kinds of cells are not as specific as is generally thought. This is especially true for neuroglial-specific stains. This is a list of elements in the central nervous system. According to the authoritative literature, Cahal's gold sublimate shows up these elements. Del Rio Ortega's stain shows up these. Bilshovsky's these. And Golgi-Cox is described as staining all these, though mysteriously only shows up 2 to 5% of the neurons.
Although most neurohistologists would agree on the identification of many cells on a particular slide, there might well be disagreement on some of the individual cells which could not be identified unequivocally. For example, phosphotungstic acid hematoxylin is said to stain astrocytes, but can we be certain whether these are neurons or astrocytes? Weil and Davenport stains microglia and oligodendroglia, but can we differentiate these two cell types here?
We approach this question in another way. We cut 5 serial sections of recognizable parts of rat brain and stained each with one of 5 specific stains. We then looked for cells which appeared in adjacent sections. Watch the position of the cells arrowed in these photomicrographs of the sections stained with hematoxylin and eosin, Marsland, Glees and Erickson, phosphotungstic acid hematoxylin, Weil and Davenport, and Gallias's stain. Having noted the number of cells appearing in adjacent sections over 8 groups of serial sections, these are some of the figures, we calculated the percentages and found that for all observations the mean was 55%. That is, 55% of cells stained with one so-called specific stain showed up with another. Another demonstration of this lack of specificity is that those most easily identifiable of neurons, the Purkinje cells in the cerebellum and the ventral horn cells in the spinal cord, show up with nearly all staining procedures. Here we see Purkinje cells stained by hematoxylin and eosin, cresal violet, phosphotungstic acid hematoxylin, and Palmgren's stain. Another example, ventral motor neurons of the spinal cord stained with hematoxylin and eosin, Del Rio Ortega's stain, Golgi stain, and cresol violet.
All these experiments lead to the conclusion that virtually all procedures, both general and those believed to be specific for neuroglial cells, also stain neurons quite clearly. In addition, the major part of the tissue is not stained by any procedure. This is contrary to the general view that, were it possible to get one section of central nervous tissue to take up all specific stains, it would appear stained almost completely and solidly. Now, unfortunately, we cannot test this directly, since one cannot stain one section with several different staining procedures and see if the addition of each, believed to be specific for each cell type, would gradually fill the whole section with a mosaic of coloured cells.
However, there are other indirect ways by which we can test the question as to whether there is any significant volume of the central nervous system not stained by all the staining procedures thought to be specific for neurons, astrocytes, oligodendrocytes, and microglia. We stain sections of frontal, parietal, and occipital lobes, cerebellum and thoracic and lumbar spinal cords, with Marsland, Glees, and Erickson stain for neurons and their processes, phosphotungstic acid hematoxylin for astrocytes, and Weil Davenport for oligodendrocytes and microglia, as well as the more general stains, hematoxylin and eosin, patase, and galliases. Using a quantimet counter, we measured separately the total areas stained by each of these staining procedures. As we have noticed before, each procedure stains structures which account for only a small percentage of the section. When we add up all the areas for these three stains, Marsland, Glees, and Erickson, phosphotungstic acid hematoxylin, and Weil and Davenport, we should obtain a value for the total area of stained tissue, as if the same section had been stained with all the different cellular procedures at once. However, only about 20% of the brain is thus accounted for. The same is true for spinal grey matter.
Therefore, one must conclude that most of the central nervous system consists of a substance which does not contain any of the elements shown up by these staining procedures, despite the general belief that virtually the whole volume of the central nervous system is filled up with neurons, astrocytes, oligodendrocytes, microglia, and ependymal cells. For example, Windle, an authority on neuroglia, said,
"There is no ground substance in the central nervous system, except in the thin basement laminae of capillaries, and the few places where connective tissue penetrates."
While Hahn, in his textbook of histology, tells us,
"In fact, there is no connective tissue space in the brain."
Nauta and Viertag, in an issue of Scientific American devoted wholly to the brain, takes this further by stating,
"If all the neurons were stained, the tissue would appear black." The idea that the brain and spinal cord are solid with cells and their processes is generally believed to be supported by observations using electron microscopy. In this example, there appears to be only a small extracellular space between identifiable elements, although one must hasten to add that this view is largely a circular argument. It starts with the belief that the brain is full of cells and their processes, and then describes the appearances between the neurons as that of the particular kinds of neuroglia. The missing point here is the demonstration by light microscopy, by which the neuroglia were originally and specifically described, that the central nervous system is indeed largely packed by neurons and neuroglia cells. Our quantimet experiment demonstrates that there is much space unaccounted for. We may then ask the question raised by Campbell in 1905. What is that terra incognita that remains over when cells, fibers, and neuroglia are subtracted? We have to approach this indirectly by defining more accurately what neuroglia is. Looking at a section of the occipital lobe of the rat, stained with hematoxylin and eosin, we see that the brain contains blood vessels and their contents, which are common to all tissues. One can also see 3 cellular elements: neurons, labelled with a capital N, some larger, more spherical paler cells, which are probably poorly stained neurons, labelled with a larger P, and neuroglial nuclei, shown by 2 small N's. And, as we have demonstrated, when one examines a rat brain stained with an astrocytic stain, phosphotungstic acid hematoxylin, one sees the same 3 elements: neurons, rounder cells, and nuclei. The same elements are seen with the triple stain of Pate and the neural process stain of Marsland, Glees and Erichsen. Let us look more closely at these neuroglial nuclei. They seem suspended in a field of material which itself takes up very little stain. Perhaps, as has been suggested for oligodendrocytes and microglia, their nuclei have only thin rims of cytoplasm. We have seen the appearance of such rims, but we ascribe them to the effect of focusing through a thick section, as used with Golgi stains, as we see here. Obviously, the nuclei are not all seen in focus in one plane, so those out of focus in any particular plane appear to have a thin rim of cytoplasm around them. If this apparent rim is an artifact, where is the cytoplasm of these nuclei? We made up several slides, each with a section of kidney, liver and brain, and stained each slide with one of several specific stains. Hematoxylin and eosin showed the cell membranes around the kidney and the liver cells, but there is no evidence at all of any such membranes between the neurons and the microglial nuclei which predominate in the section of the brain. Similarly, phosphotungstic acid hematoxylin showed membranes round the kidney and liver cells, but none between the neurons and the nuclei in the brain. This was true for the remaining stains: Violin Davenport, Grisal Violet, Gallias's stain, Pate's triple stain, and Marsland, Glees and Erichsen's. We were therefore forced to the conclusion that the nuclei, which did not appear to be surrounded by identifiable rims of cytoplasm or by cell membranes, must be either an extremely rare type of cell with a nucleus and no cytoplasm in a large amount of extracellular material, or nuclei in a shared cytoplasm, that is, a syncytium, as occurs in fungi. In order to distinguish between these 2 possibilities, we took out pieces of tissue between the neurons from random parts of the nervous system and found them to be replete with mitochondria, showing up well with Janus green, just of course as do the mitochondria in neurons. On the lower right, the mitochondria can be seen in neurons. On the left, they can be seen as clearly in neuroglia. The findings of electron microscopy are compatible with those of light microscopy. In this section, at relatively low magnification, there are 2 large neurons, here and here, with nuclei and cytoplasm, and 2 smaller spherical nuclei, not apparently surrounded by a cell membrane. Mitochondria are plentiful in the surrounding tissue. It has sometimes been argued that the neuroglia have cell membranes which cannot be seen by electron microscopy, because the section has not been cut normal to the membrane. However, this explanation cannot be accepted because the membranes around the nucleus and mitochondria are so clearly seen along most of their lengths. One can say very little about the structure of naked nuclei, since no one has succeeded in isolating them gently. In stained dehydrated section, they appear to have granular, non-uniform deposits, but no nucleoli.
We have reassessed the evidence for the current views on neuroglia, and we are now in a position to reclassify the elements of the central nervous system. All cells with processes are neurons. They include what others have called astrocytes, the astrocytic processes being the axons and the dendrites. Some neurons do not shrink or stain as much as others, so they more closely resemble the larger, more spherical, freshly dissected neurons we saw before with hematoxylin and eosin staining. We do not agree with the widely held assumption that many cells have processes which do not stain. Therefore, apart from neurons and capillary cells, the only cellular elements are those which have no processes of the naked nuclei, which other authors name reactive astrocytes, oligodendrocytes, or microglia. In part 2 of this program, which follows shortly, we shall be discussing synapses, and finally we will summarize our views on the cellular structure of the brain.
Sherrington, in the early 1890s, proposed that cells must communicate, and he suggested the physiological concept of synapses in order to interpret his experiments on reflexes. Very soon afterwards, Harold, whose illustrations we see here, observed end feet, and these gradually became accepted as the sites for synaptic activity, whose properties have been described. They are also illustrated in this silver stain preparation of anterior horn cells, produced more than half a century after Harold's drawings by Wyckoff and Young. These granules are said to be the synapses. Note the length of what must be presynaptic fibrils. We shall be returning to this point later. Synapses are also seen in electron microscopic preparations, which also involve the use of heavy metal salts. But there are several anomalies in the evidence produced from light and electron microscopy. Firstly, regarding the numbers of synapses. Each point on this diagram is plotted from figures quoted in authoritative publications and shows that by light microscopy, synapses appear in tens to hundreds on the surface of neuron bodies, while by electron microscopy they appear in their thousands. Similarly, when we compare the figures quoted for the diameters of synapses by light and electron microscopy, there is again a large difference, as we can see from this diagram. The average sizes for light microscopy are usually above 1 micron, those for electron microscopy below. We now consider the actual appearance of synapses. These drawings by de Robertis summarize microscopists' findings. This is a common view by light microscopy, showing the synaptic cleft and the pre-terminal fiber. By electron microscopy, although it is rare to see anything of the pre-terminal fiber, we often see a clear view of the synaptic cleft with its characteristic pre- and post-synaptic thickenings. The cleft has a very consistent appearance in all specimens, with the elements quite specific distances apart. This is a model of a synapse. If we were to cut a medial longitudinal section and view it, as microscopists do, at right angles to the cut, the cleft would have this appearance. If we cut at another angle and again view at right angles, we will see, relative to our first section, a much extended area of thickening, and the cleft will appear closed. It is obvious that the appearance and dimension of the cleft are going to change with the angle of the section. The lack of variety by electron microscopy is not consistent with the largely random orientation that these irregular three-dimensional objects will have to the microtome knife. It was well into the 1950s before Houdin succeeded in isolating unfixed neurons for examination. Such neurons have a granular appearance, and some histologists have interpreted this as being due to synapses. But are the granules on the surface of the cell, or are they in the cytoplasm? Here are two models of a cell. We have taken a thin section from each to show that the one on the left has granules on the surface, and the other has granules in the cytoplasm. Viewed from above, the distribution of granules is different. Those on the surface appear fairly evenly distributed, while those in the cytoplasm are less numerous over the nucleus. When one examines whole neurons by low-power microscopy, where there is considerable depth of field, or looks at illustrations of living cells in tissue cultures in the literature, a list of whose authors is shown, one sees in every case that the granules appear less crowded over the nucleus. Therefore, one must conclude that they must be in the cytoplasm, and the appearance is probably due to mitochondria in which neurons are rich. So if these granules are in the cytoplasm, they cannot be synapses, since synapses by definition should be on the surface of neurons. What then of the granules seen in microscopic preparations, stained or fixed with silver salts, which we saw earlier? Certainly they appear on the surface of neurons, but we can also see many granules in the background between cell bodies. This is also the case in Harold's drawings. This is a universal appearance by light microscopy. We list here authoritative publications which illustrate the granules sited between and away from the neurons, sometimes as frequently as on the neurons themselves. This itself casts considerable doubt on the identification of these granules as synapses.
There is an even more serious objection to regarding these granules as synapses. Although there are many illustrations in the literature of neuromuscular junctions, where can we see any evidence of synaptic connections with other neurons?
Looking at this drawing by de Robertis, we can see that the synaptic knobs are not attached to any pre-terminal fibers, dendrites, axons, or any other part of another cell body. The same is seen in Wyckoff and Young's micrograph. In some of the granules, short strands can be seen attached, and this shows that the thickness of such connections would be within the resolution of the light microscope. Indeed, in cerebral and cerebellar white matter, one can see fibers of the same diameters as these strands. This demonstrates clearly that connections with synapses could be seen if they were there. These drawings show what the neuron and pre-terminal synaptic connections would look like in plan and side views. Such connections are just not seen down the microscope or in the literature.
It is of crucial importance that if synapses are to carry out the physiological function attributed to them, they must be connected to dendrites or somas of other neurons. In the light of these conclusions, let us look again at the structure of the different cells in the central nervous system.
The neuron is thus a cell with processes, called axons or dendrites. Its soma has no synapse, boutons, knobs or end fossa on its surface. In life it is fairly spherical. The cytoplasm is a clear liquid of low viscosity with any mitochondria in it. There is no Golgi body endoplasmic reticulum or any lysosomes. The nucleus is imperforate. In unfixed neurons in normal saline, one can see a thick nucleolar membrane. The nucleolus contains a skein-like nucleolonema, which can be seen moving when the neurons are alive. The positions of the neurons, axons, and dendrites in the nervous system are fixed in life due to their ramifications. The structure of the naked nuclei is simply that of a spherical ball containing a uniform, clear nucleoplasm, in which there are no nucleoli. They move slowly in a semi-liquid neuroglia, which is a syncytium rich in mitochondria. It contains capillaries and is lined by ependymal cells along the ventricles. This diagram is similar to the micrograph of Scholl, shown here, and of other authors such as von Economo, Campbell and Connell.
We may conclude, firstly, that the central nervous system is composed of a comparatively small volume of neurons and their processes in a much larger volume of syncytial ground substance or cytoplasm in which naked nuclei and mitochondria are interspersed. Astrocytes and oligodendroglia with processes are probably neurons. Secondly, the properties of the syncytial ground substance and of the neurons dominate the biochemical, biophysical, mechanical and pathological properties of the central nervous system in vivo and in vitro. It should be stressed that the syncytium described here is located between the neurons and the naked nuclei and is quite unlike previously reported syncytia, which were continuous with intracellular fibrils as seen in this picture of Bowers. Thirdly, excitability crosses the nervous system not by chemical transmission at synapses, but by electrical conduction from a neuron body down the dendrites and through the low-resistance syncytium. This radical reappraisal of the cellular structure simplifies the explanation of a number of well-known properties of the mammalian central nervous system. The rapidity with which gliosis occurs after focal cerebral infections, the rapidity of spread and the malignancy of gliomata, the infrequency with which individual astrocytes, oligodendrocytes and microglia can be identified and distinguished unequivocally in healthy tissue, the difficulty of separating and identifying pure fractions of these kinds of cells, the fact that edema seems to be mainly neuroglial, the softness of the living brain compared with other cellular tissues, the low electrical impedance of the living brain, the fact that some patients with severe hydrocephalus and cerebral atrophy may have normal physiological and psychological behavior patterns, the high oxygen uptake of the brain and the spinal cord. The diminution and clarification of cell types in the normal central nervous system should simplify considerably the classification of pathological conditions, especially of tumors, arising in the brain and in the spinal cord.